I am going to continue with lecture 3. We are now in the regulation of genetic expression. And basically, what we have here is areas known as operons where your RNA polymerase will bind in order to start transcription of messenger RNA. Okay, these are known as inducible operons because therefore they need to be activated. And an example of an inducible operon is the lactose operon. The repressible operons are the opposite. And those are the ones that actually deactivate by represses when there is no more need to make a specific protein. So right here is a repressor, which blocks the RNA polymerase. And when you have enough of your RNA polymerase, as you can see here, is making your messenger RNA. And once that is done, the lactose will block and turn off this actual system. Here, the tryptophan gene is turned off again when it's no longer needed by a repressor. So your inducibles will happen to be catabolic, and your repressor will be anabolic. As I mentioned earlier, mutation does not necessarily mean that is a bad thing. However, it rarely leads to a good thing, which is something that we need to remember from natural selection, which is known as adaptation. There's different types of mutations, which I'm going to go over with you now. Some of them are known as point mutations. Some happen to be also known as frameshift mutations, or also we have the large scale mutations. So this is known as wild type DNA or dynamic messenger RNA. The wild type means it's normal, by the way. When you have one specific base pair mutation, as you can see here, originally it was AAA, now it's AAG. That's known as a point mutation. However, if it gives you the same exact amino acid that you see here and here, it's known as a silent mutation because nothing happens. Missense mutation happens to be a mutation that is also of one base pair. However, in this case, you can see that it does change the amino acid that you get from the original wild type. Nonsense mutation means that you get a stop codon too early. So they call it nonsense because it makes no sense that, usually your polypeptide chains will be hundreds of thousands of base pairs, and here is like a sudden stop. Frameshift would be because you have inserted an extra base, and therefore your codons are 3 at a time, so therefore the whole thing would change the information by causing a frame shift. It could be because of an insertion, or it can also be caused by a deletion. Anything that causes a mutation is known as mutagen. This could be due to radiations, it could be due to chemical mutations, mutagens like that causes your DNA to be distorted. And as you already know, this can also cause frameshift mutations. This is a mutagen known as UV light, and this causes thymine dimers. And that basically means that you have 2 thymines paired one with the other right here. This is an example of a point mutation. This is showing you how the frame shift occurs. How is this frequent as far as mutations in your DNA or DNA in any organism? Very rare. As you can see, it's 1 per like 10 to the 7th power million. So that's like, trillions, 1 to a trillion. But when these mutagens present, it does increase the factor from 10 to 10 and 1,000 times. The one that is actually more in charge of repairing your DNA happens to be DNA polymerase. Okay, mutants are the ones that are created as far as mutations. Your wild type is when you have a cell that is normal. And some of the recognizing methods of mutants, again, like I mentioned to you, it happens to be your DNA polymerases that recognizes these mutants. Here, this is diagram showing you how recombinations occur. And basically that means that a section of your DNA pairs with one another and it exchanges information. This generally also happens when it's supposed to happen, actually, is during meiosis. And when you have your DNA swapping information between the two different sister chromatids. This is known as either vertical or horizontal gene transfer. It depends how it occurs. If it's vertical, it's just from you passing your genes to the next generation, which happens during, again, meiosis. Horizontal happens in different types. You have transformations, which is when bacteria just takes up new DNA. Transduction is when you actually have the bacteria getting DNA from a bacteriophage, which is a virus. And conjugation is just when two bacteria exchange with a pili or through a pili their genetic information. This is your bacteriophage, which Hershey and Chase discovered this with the use of the bacteriophage to determine which is the part that goes into a bacteria during transduction, and it happens to be DNA. This is conjugation. As you can see, they're passing the genetic information through a pili, and this is known as a F factor, which is known as for the fertility factor. And then it generates a new pilus at the end. These are the mechanisms and the requirements for these mechanisms. And now we're going to begin with chapter 11. Characteristics and classifications of prokaryotes. What's most important in this chapter is that you have to remember that these are real prokaryotes, and the characteristics of bacteria are a more due classification based on if they are gram-positive versus gram-negatives. And in each of these groups, the amount of guanosine and cytosine that they have, if it's above 50% or below 50%. These are your morphologies. You have Coccobacillus, Bacillus vibrio, your spirillum, spirochetes, your pleomorphic, meaning that they have different shapes, and your star. Okay. Your endospores are characteristics that happen due to bad environments for the Bacillus clostridium, and then they generate these endospores in order to survive that bad environment, especially when they lack the actual nutrition. They all reproduce asexually, meaning that they actually don't need another organism. They do this through binary fission or snapping division, reproductive spores, budding, or viviparity. This is, as you remember, binary fission, very similar to mitosis. However, this is a sexual reproduction. It has nothing to do with mitosis. Snapping division means you have a newer inner portion of the cell wall generated. Generation of spores that comes from the filamentous vegetative cells. Budding, which is also a characteristic of bacteria, but however, you also find this in yeast, just like you also find spores in other fungus. Okay, these are your viviparous behaviors. Your arrangement of prokaryotic cell, I mean, how do they look by their fission? Well, this is generated by planes in which the cell divides and then they separate. These are the different arrangements of the cell for the cocci versus the bacillus. We have the prokaryotes, however. Within these prokaryotes, we have the archaeobacteria and the eubacteria, and then we also have the other organisms which are forming part of the eukaryotes. The Berger's manual was generated in order to classify all these different prokaryotes. These are the different labels used in the industry and in healthcare, of course, depending on the organism and depending on the actual pathogen. I highly recommend you to start forming maybe tables so you can start labeling which ones are low gram positives. I mean, not gram positives, excuse me, guanosine and cytosine gram positive bacteria. In instance, here you have your bacillus, which they give you different examples here. You have your clostridiums, your mycoplasms, and your listeria. More of these low G and C gram positives are your streptococcus and enterococcus and your staphylococcus. The high content of guanosine and cytosine gram positive are your corynebacterium, okay, which are aerobus, your actinomycetes, and your mycobacterium, which are known as acid fast stain positive, your mycobacterium. This is a table that I recommend that you actually look at and you use as a good studying tool. The gram-negative basically means that they lack a big pectoglycan wall, so that's why we call them gram-negatives. They're the most diverse groups, though, and specifically the gamma proteobacteria are the most diverse. Most of these organisms you probably have seen also through your laboratory. The chlamydia happens to be the most common STD causing agent of bacteria. Here's your gram-negative proteobacteria. Now they're going into different areas, and the alpha, they call it alpha because of the actual protein that they have. Again, these are your betas. And he also tells you what each of these cause as far as pathogens. This is your gamma. And they're known as glycolytic because they go through glycolysis. There are 3 different families, which is your Enterobacterus, your Vibrios, and your Pasteuroses. Very common bacteria you probably heard about, your Pseudomonas. They're infectious to the urinary tract, ear, lungs, and through your inner connective tissue under the skin. More of these gram-negatives, proteobacteria. Here we have delta proteobacteria and your epsilon proteobacteria. One of the common bacteria you might have heard is the Helicobacter, which is main bacteria that causes ulcers. Your spirochetes, these are motile, meaning they can actually move. Here's also your chlamydias. Again, like I mentioned to you earlier, it's the most common STD bacteria, not only in the United States but worldwide. Your bacteroids, they're actually probacteria because they're good for your digestive tract. They help us digest food. However, they can cause infections only when we have another type of illness, meaning that your immune system is low. Thank you very much. And this is the end of lecture 3.